Alright, welcome back, fellow scientists. So today we're going to continue our discussion of biotechnology. If you'll remember from last video, we talked about one tool and one process. The first tool was restriction enzymes, and those are enzymes that cut DNA at specific sequences. They can either produce blunt ends, everything's paired up, or sticky ends. We can have a couple of unpaired bases that want to pair with something else. And then we can use that with our first process called transformation or recombinant DNA. We can take DNA from one organism, and then using those restriction enzymes that produce sticky ends, we can splice that into DNA from another organism. So with this video, we're going to look at two more tools and two more processes. So our second tool in our discussion of biotechnology is called polymerase chain reaction. It's abbreviated PCR. And its job is it's used to make millions of identical copies of DNA, right? So you should recognize this word polymerase right here, right? DNA polymerase, RNA polymerase. Basically, it's just an enzyme that's going to make a polymer, a specific polymer, either DNA or RNA, right? DNA polymerase makes DNA, RNA polymerase makes RNA. And we know it's an enzyme because of the ACE ending. Okay, so here's how it works. We have our original DNA, obviously double helix, right? And so then the PCR machine, it's going to take that original copy of DNA and it's going to heat it. Okay, what heating does, it denatures proteins and it also can separate DNA molecules or denature DNA molecules. It can change the shape of DNA molecules. So that's going to heat it, and so that's going to separate the two chains. So we're going to have one chain like this, and then we're going to have another chain like this. And then a special polymerase that's normally found in bacteria that live in really high temperatures in hot springs. So this heat doesn't denature that protein. A really special, it's called Taq polymerase, is going to come along, and it's going to use our base pairing rules, A's and T's and C's and G's, and it's going to make a copy. Right? So now we have two copies. Well, we can repeat this cycle, right? We can heat, and so then of course what that's going to do is that's going to separate that strand and that strand, and it's also going to separate, right, that strand and that strand. And then this special Taq polymerase, which isn't denatured by this heat, is going to come along and is going to use our base pairing rules and is going to add new nucleotides. And so now every cycle of heating and then cooling, so that Taq polymerase can come along and can use our base pairing rules, every cycle is going to double the amount of copies of DNA. And remember, because A's always go with T's and C's always go with G's, every single one of these copies is the same as our original. Okay, so that's our second tool. So now that we can make millions, possibly billions of identical copies of DNA. Now, what can we do with that? Well, actually, that'll be, sorry, I forgot the order I was going in. We'll talk about that with our processes. Okay, so our third tool, go ahead and write this down, gel electrophoresis, separates molecules based on size and charge. Now, you already know this because you already did this in class, but it's good to write it down. It's good to kind of be a little bit more familiar with the process. So first thing that we did is where's my words? There's my words, is we made a gel. Now, we can make different kinds of gels, right? So one kind of gel that we can make, and we do this in AP biology and we also do this in biotechnology, is we can put the wells way over at one end of the gel. And remember, the wells are just little holes or just little indentations in the gel. Or like we did in class, we can put the wells in the middle of the gel, just like this. And those two different ways of making the gel serve two different purposes. Okay, so number one, make gel. Figure out what kind of gel we need, what kind of gel we want. Number two, put gel in chamber with salt water. Later we'll apply an electrical current, and electricity, believe it or not, doesn't flow through pure water. So we need salt water. So it's a special TAE buffer, but basically it's just salt water. Put gel in chamber with salt water, then load samples into the wells. Okay, so we'll look at this one first. So let's say that we have a negative sample here and a positive sample here, and then let's say that we have a mixed sample. We have a negative and a positive sample here, right? With this gel, we would use this gel for something called DNA fingerprinting, which is going to be our next process that we talk about, or actually our last process that we talk about. And DNA, because of the phosphate groups, are all negatively charged. So all of these are going to have a negative charge. All these samples in this well are going to have a negative charge. In class, we use different dyes with different polarities, and we were trying to determine which was positive, which was negative, and which was large and which was small. Okay, so then we're going to apply an electric current to the chamber, and so we're going to polarize. So we're going to say that this is the negative end and this is the positive end. This is the negative end and this is the positive end. Well, as you guys remember, opposites attract, right? So this negatively charged molecule is going to go towards the positive end. This positive is going to go towards the negative, and then same thing, this negative towards the positive and this positive towards the negative. So apply an electric current to the chamber, so that's going to make the dyes or the DNA move, and then we're going to analyze the results, right? So we separate molecules based on size and charge. Well, okay, Mr. Leo, so we get the charge, but what about the size? How does that work? Well, over here we have our different samples of DNA, which are all negatively charged, right? And we're going to have some samples that don't move very far, and then we're going to have other parts of the sample that do move very far, right? So now, take a guess, which ones don't move very far or move fast, the large ones or the small ones? You're right, the large ones, because typically large things move slower than small things. So large pieces of DNA will stay by the well, and then small pieces of DNA will travel farther and faster. Okay, so that is our third tool, gel electrophoresis of biotechnology. So now let's talk about some process. All right, so now let's go and let's look at our second process, DNA sequencing, which is defined as determining the order of bases in a sample of DNA, right? So up until now, chapter 12, chapter 13, we've just kind of assumed, oh yeah, there's A's and C's and T's and G's, and we can see the order because it's right there on the page. But we know that even if we use the most powerful microscope in the world, we can just barely see a double helix of DNA. And even if we could see the individual nucleotides, it's not like they're conveniently labeled for us. They're just atoms and molecules. So how do we determine the sequence of A's and T's and C's and G's? Well, the first step is to take a sample that we want to know the sequence of and make millions of copies. So use PCR, use our second tool of biotechnology. Then the next step is to add in special chain terminating nucleotides along with regular nucleotides, right? So then what DNA polymerase is going to do is it's going to normally add in these normal nucleotides, but sometimes it'll add in special chain terminating fluorescently tagged nucleotides. Okay, so let's say it has this A right here, and let's say that just randomly one of the samples, right, a special chain terminating nucleotide is added right here. And then let's go on to the next one. So let's say normal nucleotide is added right here, but then a special chain terminating nucleotide is added right here. And then for this next one, 2 normal nucleotides and then a chain terminating, 3 normal nucleotides and then a chain terminating, blah, 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 right? So then we can heat this up and we can separate the original from the newly made DNA, right, by denaturing, by heating it, and then we can take our samples and we can run them through gel electrophoresis like we just talked about. So separates molecules based on size and charge. And so this T, this fluorescently labeled T, this is going to be the smallest, and so if we load the samples here and then we apply an electric current, right, then it's going to be down here, and then this A is going to be the next smallest, and so it's going to be down here, and then the C is going to be the next smallest, so it's going to be right here. So by using normal nucleotides and special chain terminating nucleotides and millions of copies of a sample and gel electrophoresis, we can do the whole entire process of DNA sequencing, and we can figure out the exact sequence of A's and C's and T's and G's in a specific sample of DNA. Okay, so that is DNA sequencing. And then the last one that we're going to talk about, the last process that we're going to talk about is DNA fingerprinting, right? So we all know fingerprints are used by police and detectives and other people to identify people. Well, we can do pretty much the same thing with DNA. We can identify a person using their DNA, right? So the way that we do this is we take the different samples of DNA that we have. So let's say that a robbery was committed, right? And someone, I don't know, drank a soda while they were there because they were thirsty or something like that. And so left some of their saliva and left some of their skin cells on that soda. So we have DNA sample from the crime scene. Then let's say that there are 3 suspects for whatever reasons that we know. So we get a court and subpoena a sample of their DNA, right? And so then we do PCR. We get millions and millions of copies of their DNA just to make sure that we have, you know, good DNA, and we have backup copies and everything like that in case we have to do this again. And so then we use the same restriction enzyme to cut these different samples of DNA, right? Now, every single person in the whole entire world and in all of history has a different sequence of DNA, has different genes that make up their genome, right? So every single person on the planet, besides identical twins, has different DNA, right? So those restriction enzymes, they're going to cut in different places. They're going to make some long fragments and some short fragments, and it's going to be a unique mixture. It's almost like a barcode for an individual person, right? So then we can take those cut samples, those cut fragments, and we can load them into gel electrophoresis, and we can use gel electrophoresis, and we can separate the large molecules up here from the small molecules down here. And because every single person has different DNA, and because we use the exact same restriction enzyme on all the samples, then if one of the suspects was at the crime scene, then we'll be able to tell that based on their DNA fingerprint, based on their unique DNA barcode. So anyways, so that is the third process of biotechnology, DNA fingerprinting. So thanks for watching. Hope you enjoyed, and I will see you on the flip side.